This short film instructs the precise methods employed to successfully grow freshwater rainbow trout gill cells on permeable membrane inserts in primary culture through the double-seeded insert or DSI technique. This fish gill cell system can be used for studying freshwater gill physiology, toxicity testing, and for environmental water monitoring. Ensure sterile culture technique is employed, and unless stated, the following procedures take place in a laminar flow hood. Prepare the two types of cell culture media. The first is with FBS and antibiotics and is comprised of 27 ml of FBS, 11 ml of penicillin streptomycin, and 11 ml of gentamicin, all sterile filtered and added to 500 ml of L15 medium. The second is with FBS only and is comprised of 27 ml of FBS, sterile filtered and added to 500 ml of L15 medium. Now that we've made our cell culture medium, we can make the solutions ready for the dissection. A pre-washed solution, a washed solution, a trypsin solution, stop solution, and the rinse solution. The pre-washed solution is comprised of 20 ml of PBS in a Petri dish. The washed solution is composed of 1.2 ml of penicillin streptomycin, 1.2 ml of gentamicin, and 90 microliters of fungazone solution, all made to 30 ml in PBS. 10 ml of this is added to a Petri dish, whilst the remaining 20 ml is split between two 50 ml falcon tubes. Trypsin solution is composed of 650 microliters of 0.5% trypsin EDTA made up to 6.5 ml in PBS in a 50 ml conical falcon tube. Stop solution is composed of 2 ml of FBS made to 20 ml with PBS in a 50 ml conical falcon tube. Rinse solution is composed of 0.5 ml of FBS made to 20 ml with PBS in a 50 ml conical falcon tube. These can all be stored on ice. Firstly, condition the permeable membrane inserts for at least 1 hour. We advise the use of 12-well inserts with a 0.4 micron pore made of polyethylene tetraphthalate or PET membrane purchased from Corning. Place the inserts at the companion wells and add 200 microliters of cell culture medium with FBS and antibiotics in the apical compartment. You are now ready to euthanize a fish according to the local ethical procedures. Afterwards, the key point is to remove the head as quickly as possible to avoid hemorrhage to the gill. This can be seen as bleeding and clotting on the filaments, and if this is seen, the gill should not be used. Outside the laminar hood, dissect out the 8 intact gill arches and place in the pre-washed solution in the Petri dish. Remove the mucus by careful blotting onto tissue and do not let the filaments dry out. Dissect them from the arches, taking care not to include any cartilage. Place them in 10 ml of washed solution in a Petri dish. Tease the filaments apart using tweezers and a scalpel, like this. Pour away the washed solution and add the filaments to a new 10 ml washed solution in a 50 ml conical centrifuge tube on ice. Incubate for 10 minutes. From here on, sterile culture technique is employed and the procedures should take place in a laminar flow hood. Centrifuge for 4 minutes at 250 G at 4 degrees C. Aspirate the supernatant. Add the remaining 10 ml of washed solution, mix well, and incubate on ice for a further 10 minutes. Centrifuge for 4 minutes at 250 G at 4 degrees C and aspirate the supernatant. Add 500 microliters of trypsin solution and agitate. Centrifuge again on the same settings and aspirate the supernatant. This begins the tryptic digestion. Add 3 ml of trypsin solution and shake for 12 minutes at 200 RPM at room temperature. Meanwhile, remove the tip of a 1000 microliter pipette tip to widen the aperture and store in ethanol to sterilize. Using these tips, pass the filaments repeatedly, at least 50 times up and down, to mechanically agitate the filaments. Transfer the whole solution and the filaments through a 100 micron cell strainer into a 50 ml conical centrifuge tube containing 20 ml of stop solution on ice. Agitate the filaments with a cut pipette tip. Remove the filaments from the strainer and replace back in the Falcon tube with 3 ml of trypsin solution and shake again for 12 minutes at 200 rpm at room temperature. Transfer again through a new 100 micron cell strainer into the same 50 ml conical centrifuge tube containing 20 ml of stop solution and the previously trypsinized cells on ice. Mix well. Centrifuge the cell suspension for 10 minutes at 250 G at 4 degrees C. Aspirate the supernatant as before. Resuspend the pellet well in 2 ml of rinse solution and then add the remaining 18 ml. Agitate and centrifuge for 10 minutes at 250 G at 4 degrees C. Aspirate the supernatant. Once you've aspirated, the pellet should look red and meaty. To get a really good cell resuspension, flick the pellet vigorously. Resuspend in 2 ml and then add the remaining 8 ml to bring to a final volume of 10 ml and mix well. Now you can count the cells using a hemocytometer. Add 10 microliters of cell suspension to 90 microliters of trypan blue solution in an Eppendorf and mix thoroughly. Quantify the concentration of cells by applying 10 microliters of this mixture onto one side of the hemocytometer. The percentage of dead cells must not exceed 5%. Count the viable cells and adjust the cell counts to the required seeding density. Now you can aspirate the medium conditioning the cell culture inserts from before. When you're ready to seed the cells, we recommend seeding 1.2 million cells in 800 microliters of cell culture medium with FBS and antibiotics per insert. To provide nourishment to the cells seeded in the apical compartment, place 1 ml of cell culture medium with FBS and antibiotics into the basolateral compartment. To maintain the hydrostatic pressure on top of the cells, it's important to always fill the apical compartment first and then the basolateral. Now you can leave these at 18 degrees overnight. This is termed day 0. Now, after 24 hours, begin a new gill cell preparation using another fish. This provides the cells to seed on top of the previous day's seed. Repeat all of these steps again, resulting in a new cell culture suspension at the same seeding density of 1.2 million cells per 800 microliters. To prepare the previously seeded inserts for double seeding, wash the inserts carefully and slowly, twice with 200 microliters of PBS. If needs be, a further PBS wash can be done if the inserts appear to have a lot of mucus on. Remember, it's critical to aspirate the basolateral side first and then the apical before washing the apical. This maintains the hydrostatic pressure so that the cells adhered to the insert are not lifted off. Apply the new gill cell preparation directly on top of the previously seeded inserts. Again, seed 1.2 million cells in 800 microliters. As before, apply 1 ml of cell culture medium with FBS and antibiotics into the basolateral compartment. Store these at 18 degrees C. This is termed day 1. 24 hours later, wash the inserts again as previously. Replace with 1.5 ml of cell culture medium with FBS and antibiotics in the apical compartment and 2 ml in the basolateral, and store at 18 degrees C. This is termed day 2. On day 4, change the media to cell culture medium with FBS so that antibiotics are no longer present. From this point onwards, a daily measurement of transepithelial resistance, or TER, can be made using a custom-modified epithelial tissue voltometer fitted with chopstick electrodes. A blank insert for 12 wells will give a value of approximately 180 ohms per centimetre squared. Once blank corrected, a reading of 70 to 120 ohms per centimetre squared is encouraging. At around day 7, DSI epithelia should show a TER value of approximately 5 to 10,000 ohms per centimetre squared. It's important to know that the TER may develop more slowly in some preparations, in which case, keep monitoring them daily. Inserts which have not reached 5,000 ohms per centimetre squared by day 14 may not develop and should not be used. Above a TER value of 5,000 ohms per centimetre squared, fresh water can be applied to the apical surface of the cells. Typically, this application of fresh water will cause a rise in TER. Importantly, after freshwater application, inserts around 5,000 ohms per centimetre squared may rise to 30,000, whilst those already around 25 to 30,000 may not rise by as much, to perhaps around 35,000. After freshwater applications, TER values should remain high for between 24 to 48 hours. To prepare for freshwater application and experimental procedures, it's crucial to aspirate the basolateral compartment first and then the apical. To wash, apply 800 microlitres of PBS in the apical and then 1000 microlitres into the basolateral compartment. Aspirate the basolateral compartment and wash a further 2 times before the final aspiration. With an empty basolateral, you can now wash the apical compartment 3 times with 800 microlitres of PBS. Apply 1.5 ml of fresh water, or whatever the test protocol requires, to the apical compartment, whilst leaving the basolateral bathed in 2 ml of L15 medium without FBS. I'm Lucy Stott, and I'm a BBSRC case student with AstraZeneca at King's College London, and this project was funded by BBSRC Sparking Impact.